between those sequence types. So this is quite a broad lethality model. We thought maybe the difference between ST313 and ST19 might be a little more subtle. So what we decided to do was to look at dissemination. So we infected mice perorally with either I77, which is an ST19 strain, or D65, which is an ST313 strain. And then we sampled the spleen, liver, and blood at different time points, so 3 hours, 8 hours, 24 hours, and 72 hours post-infection. So firstly, there was absolutely no difference in the organ counts. Both ST19 and ST313 were able to access the spleen and liver at equal levels. However, we did see a significant difference in the blood counts at 24 hours post-infection. So you can see here on this graph showing CFU per mil of blood, and we have ST313 in the orange and ST19 in the blue, that we did have more bacterial burden in the blood at this 24-hour time point in both CD1 and BALB/c mice. Interestingly, we don't see this trend at any of the other time points, so it does look to be a very transient increase in invasion. So while the mouse model did give us some small amount of information about invasion, we really wanted to take this further and look at a much more relevant model of infection. And so we moved to the rhesus macaque infection model. So rhesus macaque studies were first carried out back in the 1960s and 1970s. And what they showed was that animals infected with Salmonella typhimurium had diarrhea, and that they also showed signs of severe intestinal inflammation. So we actually recently revived this model to investigate vaccine safety of our live attenuated Salmonella typhimurium CVD 1921 strain in both SIV positive and healthy rhesus macaques. And when we did that study, we saw quite similar symptoms in those rhesus macaques to humans, including diarrhea, fever, lethargy, weight loss, and also intestinal inflammation. So to answer the question about ST19 and ST313 in monkeys, we chose 6 Indian rhesus macaques that were Salmonella negative, 3 per group, and we challenged them intragastrically with I77, our ST19 strain, or D65, the ST313 strain. We then monitored the monkeys for 3 weeks for signs of infection, including several clinical signs such as temperature, weight loss, and white blood cell count. We looked at the bacterial load in stool, we did stool grading, we did some blood culture, and at necropsy at 3 weeks post-infection, we also did histopathology and looked at bacterial counts in organs. So in the interest of time, these are the things I'm not going to show you. So we did look at clinical signs, however, we didn't see any difference between the 2 sequence types. So there was no difference in temperature, weight loss, or white blood cell count. We also did blood culture, but we weren't particularly successful at getting positives. So we took blood at 1, 3, and 15 days post-infection and looked for bacteria in the blood. We only had 2 positives over that time, one from an ST19 infected macaque and one from an ST313 infected macaque. And we think the problem for this is really sampling, because these are quite small monkeys. We can only take around 1 ml of blood to do culture from each time. And finally, we didn't see any difference in bacterial counts in the organs, and we think that's probably because it was 3 weeks post-infection, and by that time the bacteria were quite well cleared from the macaques. So now on to the more interesting stuff. What did we find? So we first did stool counts every day of the infection to determine the intestinal burden. And so you can see here on this graph showing CFU per gram of feces. We have the ST19 strains in blue and the ST313 strains in orange. And you can see that at both days 3 and 4 post-infection, we had significantly more bacteria in the ST19 infected macaques than we did with ST313. In addition, you can also see that the ST19 bacteria sort of hung around for between 11 and 21 days post-infection, whilst those infected with ST313 cleared the infection within 5 to 11 days. When it came to stool grade, we actually scored the diarrhea severity every day. 1 was considered mild, so loose stools, 2 was moderate, and 3 was severe, like dysentery. So if you see here the cumulative diarrhea score for ST19 in blue versus ST313 in orange, you can see that ST19 infected rhesus macaques had significantly more severe diarrhea than ST313, especially on days 3 and 4, where all animals in our ST19 group had severe diarrhea, whereas no ST313 infected rhesus macaque showed moderate or severe diarrhea at any time during the study. Finally, moving on to the histology. So this was done on the organs at necropsy, so 21 days post-infection. And what you can see here is some histology from the liver, colon, ileum, and mesenteric lymph nodes. We also did the spleen, however, we didn't see any significant differences. So here you can see that ST19 infected rhesus macaques showed significantly higher levels of inflammation in these organs compared to ST313 infected rhesus macaques, which showed basically normal tissue. So to summarize this rhesus macaque data, ST19 strain I77 induced significant levels of diarrhea in infected animals. They also showed very high levels of intestinal inflammation, and they had much higher shedding than the ST313. So ST313 strain D65 causes less diarrhea. It shows decreased colonization of the intestines and reduced shedding, and it also induces less histopathology in organs than ST19. So to conclude, the mouse model is quite limited in its ability to model virulence in Salmonella typhimurium ST313. Although we did see a very small increase in virulence, in invasion at 24 hours, this is quite a minimal difference. However, the rhesus macaques are a very, very good model for Salmonella typhimurium-induced gastroenteritis. They show many of the same signs that we see in humans, including weight loss, lethargy, diarrhea. And we've shown with this model that, as people have postulated from the epidemiological studies, that ST313 strains seem to be less able to cause diarrhea in this model of rhesus macaque infection. We weren't able to answer the question of whether ST313 are inherently more invasive than ST19 in this model, and we think that in the future we could potentially look into that by using earlier time points. So we think if we could take larger blood volumes, and also if we could look in the organs in the first week of infection, we may be able to see a difference between ST19 and ST313. So there's quite a few people I need to thank for this work. So firstly, Dr. Girish Ramachandran. He was the person that really started all of this ST313 pathogenesis stuff, and he's now at the FDA. All the mouse work I showed was his. Dr. Aruna Panda is a veterinarian that's in charge of all our rhesus macaque experiments, and all this work was done in the lab of Dr. Sharon Tennant. I also need to thank the clinical microbiology team, who do all of our stool counts for us on a daily basis for over a month, and also the comparative medicine team, my team of vets that helped me with all these rhesus macaque experiments, and our funding came from the NIH.
Okay, so this paper is now open for discussion. Has anyone got any questions? Go ahead.
Rob Heidemann from UCL. I mean, if this is a good model of human disease, then you wouldn't expect these adult monkeys who are presumably immunocompetent to get invasive disease. Have you tried it in your SIV model, which I guess would be closer to what happens in adult humans?
Okay, so the monkeys we used in the SIV infection were actually much older than the ones we're using currently. As it turns out, it's quite difficult to get aged monkeys at this time in the US due to Ebola and Zika. So these monkeys are actually quite small, usually 1 to 2 years of age. So we're looking at quite juvenile monkeys.
But what's that in human years?
I have no idea what that equates to, but probably something similar.
Okay, thank you.
Mike.
Rob asking a question stimulated a synapse. Actually, I think you have data that are a hint to support an important set of differences of 313. You had less shedding. You had that early blip, very short, but it was very significant blip of bacteremia. And this reminds me of what one sees with type 1 pneumococcus, which is the most virulent of all pneumococcal serotypes. When you do prevalence surveys, you don't find it. And the way it behaves is when it comes in, it immediately invades, goes to bad places, and causes bad disease. I think you have some data that are supportive of that.
Thanks, Mike. Any other questions? Duke?
No, this is...
Say your name, please.
I'm Gordon Dugan.
Your microphone's not on.
Oh, that's. What's the point in asking me to say my name if I'm, yeah. Anyway, this is really interesting for me because when we first looked at the genome of ST313, you could predict from the genome that you have less shedding and less diarrhea because of the effectors knocked out, etc. And so one of the other signatures of ST313 is it's human adapted in the sense of transmission. It's not going through a zoonotic source. So, but I'm just wondering how we can get a handle on that. At the moment, I agree with the comment that Rob made that if we had an immunocompromised host, then that would be an opportunity. Are you planning to do that in the future in terms of the immunocompromised host?
So all those experiments that we did on the immunocompromised SIV-infected rhesus macaques were done at the NIH with Srini Rao, who has unfortunately left the NIH, and so we're unable to get access to those SIV-infected rhesus macaques any longer.
But also, this does validate Sam Karuki's initial hypothesis again that these are the transmission is probably more human to human than it is zoonotic to human.
Yeah, definitely.
We only have 1 more minute for a question.
Well, just to say that Duke's right. That paper by all the SIV infected, when they got the salmonella, they had rather devastating disease, and a couple had to be put down.
Yes, that's true.
When they got the wild type.
Yes, SIV-infected rhesus macaques that were given wild-type I-77, but this is ST19, not ST313.
So there's just one other experiment you should do. These are also multi-drug resistant, right? So there's probably competition with the microbiotas. Do you pre-treat your monkeys with antibiotics?
No, they're not pre-treated.
So if you pre-treat with antibiotics, I would hedge bet that you will get invasion and your ST313s will go in more than the intestine.
That would be a very interesting study for us
Try
to
and do that.
Okay.
Thanks, Duke. And Mike. And Rob.
Wonderful. Thank you very much.
Okay, so our next paper comes from Tahir Yousufzai from the Aga Khan University. And his talk's going to be on outbreak investigation and assessment of risk factors of ceftriaxone-resistant S. typhi from Hyderabad in Pakistan.
Hello, good afternoon. I'm Tahir Yousufzai from Aga Khan University, and in the next 10 minutes I'll be going to present the first ever, one of the largest outbreak investigation of ceftriaxone resistant typhoid fever in Hyderabad city of Pakistan. So the outline of my talk is, initially I'll be giving you a brief background of this outbreak, followed by the preliminary finding of this outbreak, and then I'm going to tell you a little bit about the control measures of this outbreak. And since, as I speak, the outbreak is still going on, so I'll also share with you that what is the way forward regarding this outbreak. So on November 30, 2016, a clinical laboratory of Aga Khan University Hospital reported a case of a child admitted to the Aga Khan Maternal and Child Center, Hyderabad, with ceftriaxone resistant S. typhi. And Aga Khan Maternal and Child Center, this is one of the secondary care hospital of the Aga Khan Hospital, which mainly cater to the middle class clientele. The microbiologist was alerted by this unusual susceptibility, thinking that it is misclassified as S. typhi rather than INTS, and they went for repeated identification and MIC. And I would also like to share with you that the Aga Khan University Clinical Laboratory is actually CAPE certified, so that has to be kept in mind. About the MIC, identification of typhi was confirmed with the API 20E and serology as well. MIC were performed on Vitek2, and it shows resistant to ampicillin, cotrimoxazole, and ciprofloxacin with high MIC. Ceftriaxone MIC was of more than 64 microgram per ml, and there was susceptibility to imipenem, meropenem, and azithromycin. And this was not only for these initial cases, but as well for all the other cases which I am going to report today. So within a week, 2 more cases were identified from the same hospital, from the same area, and as part of a usual reporting system, alarm was raised, and the government officials were informed about this outbreak. We formulated an outbreak investigation team. As you see the picture, in the first picture, there was a meeting with the Director General of Health, Hyderabad. And then in the second picture below, you can see the team with the field epidemiology laboratory training program, which is actually a CDC approach in different countries. And the fellows, they were the part of our team for this outbreak investigation. So let's discuss the preliminary findings. To date, 113 blood culture confirmed ceftriaxone resistant Typhoid cases have been identified since November 30, 2016 from 2 talukas in Hyderabad. And let me tell you, taluka is a sub-district with a population of about like 1 taluka maybe, depending upon the population density, of course, but the population ranges from 2.5 to 3 lakhs to 4-5 lakhs. So there were 2 talukas specifically from the Hyderabad, because Hyderabad has 4 talukas, and only 2 talukas are the areas from where the cases were being identified, all these 113. The investigation is still underway, and cases continue to be identified. And let me also share with you that these 113 cases are actually only those cases where the blood culture was performed. And as you all know, in the developing countries, that most of the cases are being treated empirically without going for blood culture and sensitivity. So this is just the tip of iceberg, and we don't know that how much is the total outbreak. This is a figure which shows the epidemic curve week-wise from November 30, 2016 to 28 March 2017, and you can see here bumpy picture. So still the outbreak is continued, and on weekly basis we are getting number of cases. This is another figure which shows the age distribution of the cases, and you can see here that so the orange line, it shows the percentages of the cases being identified till now, and you can see here that about 90% of the cases are less than 10 years of age. And apart from this, we also received few cases above 20 years of age as well. So about the clinical features, about 60% of the cases were admitted to the hospital and treated there, and 40% were treated on outpatient basis. All of the patients recovered, luckily, and there were no mortality observed to date in our outbreak investigation, at least for these cases which we are reporting here. So this is the geospatial mapping of the cases. And let me tell you that as of today, we have only visited 38 cases, not all 115 cases, but still the outbreak investigation is going on. But let me tell you that we have retrieved basic information of all these 113 cases, and especially the complete addresses of these cases. And here you can see here in the map the pink area. So majority of the cases are from these areas, and these are actually two talukas of Hyderabad. So most of these 113 cases are living in this area, not only these 38, but all 113. And one thing more, majority of these cases belong to middle income families. So Qasimabad Taluka and the other Latifabad sub-district. So the total population is about 0.9 million, and these are not urban slums. These are actually middle income, lower middle income families are living in these areas. So that has to be kept in mind. Now this is another map which shows, here you can see in these stars, these are the number of cases. And you can see here clustering of the cases in different areas. So in the next map, so these are overall 38 cases only. We haven't received the GIS coordinates for the rest of the cases, although we have the addresses and we're going to map it soon. So these are the overall 38 cases. Now I'm going to show you one taluka. So that will be a close-up of the cases, that how these cases are clustered together. So these are the 28 cases, and you can see here the clustering. Here these pink lines, they are showing the sewerage lines, tertiary and primary sewerage lines, and you can appreciate that most of these cases are actually located very closer to the sewerage lines. And I'll be later on showing you a few pictures that there is construction going on, and in these areas especially, and most of the sewerage lines are broken down, and even the sewer and the water lines, they are in parallel. So let me tell you about the current activities. Surveillance is going on for ceftriaxone resistant typhi at Aga Khan University Laboratory, Civil Hospital Laboratory, Betai Hospital, which is another private hospital in the same area. And we are doing age-match case control study, one case with a ratio of 4 controls in this area. Other investigations are ongoing, clinical features, household water cultures, other source searching, and Sangar's sequencing of 80 isolates. We have talked to the Sangar Institute of Wellcome Trust. So they're going to do the full genome sequencing of the 80 isolates. So this is the field activities where one of our team member is going to the households and doing the investigation. These are some of the pictures which shows that how the sewerage lines, as I already mentioned, that most of the roads are being digged out for recarpeting or construction. So this is the situation in this area. And here you can see the green pipe. This is a water pipe. Here it is broken. And this is the sewerage water on the streets. So other outbreak control efforts, as you already know, that there is no standard evidence-based treatment guideline available for ceftriaxone resistant typhoid fever, apart from one report from Nepal where they have successfully used carbapenem for ceftriaxone resistant typhoid. So mostly our treatment modality is based on either expert opinion or recommendation. So azithro for outpatient, carbapenem for inpatient, and there were few patients who received both, like 4 to 5 days of carbapenem followed by 5 to 7 days of azithromycin. And you can also see here the flyer. So this was one of the effort of our outbreak control, where we make this flyer in the local language, Urdu and Sindhi, and we distributed it in the community to create awareness to control the outbreak. Apart from this, we have talked to the Bharat Biotech, and we are thankful to them. They have actually agreed to donate TCV vaccine to control this outbreak. And we are also thankful to our Drug Regulatory Authority of Pakistan, because this vaccine is not licensed in Pakistan, but as a special case, they have provided us a letter of importation. So we are waiting to ship to receive the vaccine in Pakistan. So what are the next steps? As you already know, that this is one of the largest, I would say this is the largest outbreak of ceftriaxone-resistant typhoid fever, at least to our knowledge, and still the outbreak is going on. And our preliminary case control study, it doesn't show any conclusive evidence about the transmission mechanism of this outbreak. So we are actually very interested to see that, to get some more knowledge about the transmission mechanism and also about this syndrome. So these are some of the next steps. We want to go for molecular detection of possible environmental reservoir for eliciting this transmission pathway, impact assessment of TCV vaccination and control of ceftriaxone-resistant STIFE, chronic carriage among cases of ceftriaxone-resistant isolate versus sensitive isolates, and Sanipath approach to fecal exposure assessment as presented by Dr. Christine Move from University of Emory. So we would also like to use this approach for the assessment of the transmission. And at the end, I would like to acknowledge my team from Aga Khan University, and then Bharat Biotech for providing us the vaccines, and CDC FLTP program and Department of Health, SIN, and of course the expert panel composed of Stephen Luby, Denise Garrett, and Gordon from Sanger Institute, Christine Move from Emory University. Thank you very much.
Thanks, Tahir. One question, Chris? You were up first.
Chris Parry, Liverpool School of Tropical Medicine. You didn't mention about the carbapenem being used, but I wondered, A, whether you've tested against ertapenem. I'm aware of at least one case in India, ceftriaxone resistance being treated with ertapenem, which can be given once daily, and also whether you've looked at mesalenam. So mesalenam was a drug that was tested for typhoid in the 80s and was found to be reasonably effective, and many E. coli that are ESBL positive are sensitive to mesalenam, so that might be an additional option.
Okay.
Thank you. Thank you, Tahir.
Can I respond to that very briefly? Dr. Perry, no, we didn't actually test for ertapenem. At least we haven't yet. And we also have not tested for mesalenam. But we do intend to test for ertapenem, and we'll have the MICs once more.
Thank you very much.
So next we have Senjuti Saha from the Child Health Research Foundation at Dhaka Shishu Hospital in Bangladesh.
That was pretty good pronunciation of my name, Dick. Thank you. Okay. So today I'm going to talk to you. Sorry, the time.
I've got the title.
Okay. So today I'll talk to you a little bit about the data that we obtained to show that ongoing surveillance systems can be leveraged for enteric fever surveillance. And the data comes from 2 urban hospitals in Bangladesh, and the surveillance was done for 5 years. So I think in the last 2.5 days of presentations and discussions, it has become pretty clear that one of the main, that there's a general lack of comprehensive surveillance systems for enteric fever. And this is unlike diseases like pneumonia, meningitis, and sepsis. And one of the reasons probably for that is that unlike those diseases, enteric fever usually causes infections in low-income countries, in communities and populations that have several competing priorities. And establishing a comprehensive surveillance system in those communities can be very expensive, and sustaining such surveillance systems is almost impossible without extensive donor support. Now, having said that, WHO runs a global surveillance network called Invasive Bacterial Vaccine Preventable Disease Surveillance Network, or IBVPD. The surveillance network is run in 61 different countries, consisting of 178 sites, and it specifically probes for meningitis, sepsis, and pneumonia in under 5 children, collects data on meningococcus, pneumococcus, and Hib. Now, the overarching objectives of this surveillance system is to collect epi data, monitor vaccine impact data after introduction, and characterize the circulating bacterial types and study their antimicrobial resistant patterns, etc. Now, you must have noted by now that enteric fever is not a part of this vaccine preventable disease surveillance. Bangladesh operates 4 of the 178 sentinel sites of WHO. These are high-performing sites that produce great laboratory data. Epidemiology data generated by these labs have been used, have strongly facilitated introduction of pneumococcal and Hib vaccines in the country. And I'm showing you a picture, a screenshot actually, from the latest WHO IBVPD bulletin, where Bangladesh was presented as a surveillance success story to the rest of the world. Now, so that's great, right? We collect great data on pneumococcus, meninge, and Hib. However, when we look at our data, none of those organisms is the predominant organism detected in our surveillance, especially in bloodstream infections. So here's our data from 2011. We tested blood specimens from 1,471 suspected meningitis, pneumococcus, and sepsis cases, and of them, 128 were culture positive, and 66% or 84 were either typhi or paratyphi, so enteric fever cases. And this is how Salmonella typhi compares to the rest of the organisms, despite the fact that we were not even probing for enteric fever. So seeing this, we asked the question that if we are already isolating so many typhi and paratyphi cases, can we leverage this system to make it more comprehensive and collect more data on enteric fever? We answered this question really simply. So in addition to the inclusion criteria defined by WHO to capture those meningitis and sepsis cases, we added the inclusion criteria of fever of 102 degrees for at least 3 days. Enrolled cases meant we could get blood specimen from them. And the IBVPD platform in Bangladesh is in the inpatient departments of pediatric hospitals. And because we were using this platform, all cases were under 5. And the surveillance was conducted for 5 years, 2012 to 2016, in 2 urban pediatric hospitals of Dhaka. I almost described the surveillance, this expanded surveillance system, as 2 arms of the IBVPD platform. In green is the original IBVPD platform that WHO set out to do, and in orange is the enteric fever platform that we added to it. So in the original platform, we tested 5,185 blood cultures from those eligible cases. 171 or 3% of them were culture positive, and 55% of those cases were confirmed enteric fever cases. In addition to those, with the additional arm, we collected another 1,699 blood specimens. 21% of them were culture positive, and 97% of those were typhi or paratyphi, confirmed enteric fever cases. Now, because this was the first time we were doing this, we wanted to see if we were missing cases with this inclusion criteria. So the microbiology labs of these 2 hospitals test all the blood cultures that come into the hospital. So we did this retrospective surveillance, and we found that an additional 28 cases were found through blood culture in the lab that escaped our net of the surveillance system, which means in the total of 471 confirmed cases between 2012 and 2016, in under 5, our platform could capture 443 cases. That translates to a capture rate of 94%. Now, we went on a little further because this platform only allows for capturing under 5 cases. We wanted to see what's going on in over 5 cases. So we used the same retrospective laboratory surveillance and found 283 confirmed cases in over 5 children. So now, in total, in all ages, we found 754 confirmed enteric fever cases, 62% of these were under 5, 38% of those were over 5. It's, under 5. So, and the total cost required to integrate this enteric fever surveillance into the IBVPD platform in 2 sites was about $45,000 per year. We also went on to look at disease patterns of all the isolates that we obtained, and very similar to many other studies from the country, the region, including population-based surveillances, we saw that more than 20% of the cases were coming from under 2 children. And as I said before, 62% of the cases were from under 5. We also looked at antibiotic susceptibility patterns. Again, similar to other studies, MDR rates in type V strains was about 25%, no MDR paratype V strains. However, almost every type V and paratype V strains shows decreased Cipro susceptibility. This is a great surveillance. However, there are, of course, some limitations. And one of the obvious limitations is that it was an inpatient-based surveillance. As Andrew Pollard pointed out, and we have also seen in our previous studies, that most of the typhoid cases are actually outpatient department cases. So we are definitely missing a huge number of cases. And embedded into that limitation is the fact that because it's not a population-based study, we don't have a denominator, and we cannot calculate incidence. However, we are actually adapting Steve Luby's hybrid model that was beautifully explained by Alex day before yesterday. We have defined our catchment area. We are carrying out our community surveillance right now to figure out incidence. We're also following up all those cases to understand case fatality rate and severity. So if you didn't follow anything I said or you slept through it, there's one message I want you to take home. IBVPD platforms conducted by WHO can be leveraged for enteric fever surveillance. It's quite easy to do and quite cost effective. And the data you collect from there fulfills all the objectives that WHO set out to do for all the other diseases. So with typhoid vaccines, vaccine introductions in pipeline, we strongly recommend WHO to take enteric fever under the umbrella of IBVPD. We believe this will really facilitate rapid generation of baseline data so that we can monitor vaccine impact after their introduction. Now, we know as a group that coming here and presenting data or publishing in high-impact journals isn't enough to change policies. So our team in CHRF has been involved in advocacy for decades, well, at least a decade. This is the rally. These are pictures of rallies that we organize every year for World Pneumonia Day. And really, the hope is soon we will have a World Typhoid Day, and we'll be flying green and white balloons. With that, I would like to thank you on behalf of the entire team. This is all teamwork. We are very grateful to the CAT committee for letting us come present and share our data. Thank you so much.
Well done, Senjuti. Now, have we got any questions? John, are you going to ask whether there was cases in that 2 to 11 month that were at the 2-month end?
Yes, I mean no. So we did not find any cases under 2 months. Everything was over 2.
But were they in the 2 to 6 months, or were they predominantly at the back end?
Oh, were they predominantly over 6? I think it was pretty much an equal distribution.
Okay.
I think.
Other questions?
It was such a clear talk.
That's great. It was fantastic. Thank you so much. That was wonderful. Our next speaker is Melita Gordon from the University of Liverpool, who's going to discuss a case control investigation into the household distribution of invasive salmonella in Blantyre, Malawi. Thank you, Melita.
Thank you very much. Thank you for inviting us to speak. I'm speaking on behalf of Ranesh Prakash, Ranesh Prakash, who did most of the work. So this is a study about the reservoirs of invasive NTS around which there's a large knowledge gap. We don't really understand from the genomes what the functional implications of the adaptations we see in ST3 and other invasive strains are. We know that Sam Kariuki in Kenya quite some time ago now did a study looking at index invasive cases and taking a case control approach, in which he found that using a pulse field gel electrophoresis method, he could find a significant similarity with family contacts, but there was relatively little sampling of animal isolates in that study. We also know from the Gambia in a study linked to GEMS, so this is diarrheal cases, that they linked NTS cases and controls of diarrheal disease and looked for a linkage with animal NTS isolates using MLST and serotyping, and found almost no isolate sequence type overlap between human and animal disease. We know in Malawi, very much in keeping with the GEMS data that was just presented, that if you take healthy children and sample them regularly, they will carry salmonella from short periods of time. Tony Nyirenda has found that 30% or more will have stool culturable salmonella, and more if you look by molecular methods. And we know that about half of these are Salmonella typhimurium ST313. We had an international consensus meeting in Blantyre on invasive salmonella disease, and one of the areas that was highlighted as a priority was to try and understand reservoirs and transmission. And so we set out to do this case control study to identify household shedding and reservoirs. The particular way in which we wanted to strengthen the evidence was getting comprehensive sampling, if we could, from humans and animals and the environment in the household, and using much more high-resolution whole genome sequencing approaches to inform strategy. This is our recruitment and sampling methods. We recruited index cases of invasive disease, both typhi and NTS, because we have both in Blantyre, aiming at 60 cases of each. We then visited their household within 2 weeks, selected a control household by spinning a bottle and walking 100 yards, obtained consents from both households, and then sampled at both homes from all household and family members, any animals associated with the household, and from the environment. And then we took the samples back to the lab for culture identification and whole genome sequencing. These are not really intended as comparative data. Franziska Olgemoller has shown a much bigger set of information about the households on her poster, which I would urge you to go. This is really just to describe the households to you. So the households have about 4 to 6 members per household. Only a quarter of households keep any animals, and they tended, obviously because it's an urban setting, not to be very large numbers of animals, so we were keen to sample as many as we can. People are in fairly simple households with mud walls, sometimes plastered over. Just to highlight that people are using fairly unimproved pit latrines, and that these pit latrines are usually shared by up to 4 or 5 families typically. And people have a very mixed water supply to their house. And again, I would encourage you to go to Fran's poster to look at larger numbers and get more comprehensive messages from these kind of household data. This is a picture of the sort of sites that we would be sampling for. We sampled stool, human, animal, and animals' stools. We sampled the environment using sterile boot socks by a standardized method, and we also collected water and food samples. And these are our microbiological methods. Human samples were done in selenite broth placed onto XLD. Environmental and water samples had enrichment phases and then went on to selective plates. Colonies are identified by standard means and went for full sequencing. We collected 1,500 samples over the course of this. 2 things to notice from this slide, just that we sampled equally from case households and control households, and that we were unsuccessful in growing salmonella from any food or water samples, so we're not going to discuss those further. Second thing to notice is that the numbers that we sampled from humans and animals represents the number that were available, and we sampled in equal proportion, so there was no bias there from the samples that were available. And the second is that our isolation rate from human and animal samples was approximately the same. It was around 3 or 4% from both types of samples. I've broken it down here just briefly for Salmonella typhi, and you can see again fairly equal isolation rate across human and animals for the case and control households. And by NTS, where perhaps slightly surprisingly, we didn't grow much from the households from cases or controls. The only place we found isolates was in the households of human stools, and that was very influenced by one family. Oh, sorry, we seem to jump on there. So just to summarize, as I said, a 3% overall isolation rate, 4% from humans, 6% from animals, 5% from the soil. This is the phylogenetic tree that we produce by Illumina sequencing, and it's been constructed using Enterobase. And we've left the Salmonella typhi out because they're quite outliers, and we all know that we have H58. All the invasive strains are up here at the top. They're all Salmonella typhimurium ST13, and we're going to consider those in a minute. For the moment, we're going to concentrate on the bottom half, which is where most of our boot socks environmental and family member isolates came from. So this is the bottom half of the tree, and what I've plotted on here for you are those environmental samples that we got from the boot socks from different locations all around the house. They tended to cluster in two areas around the tree, in the sort of pale pinky isolate, which we think is related to Diarizoni, and then up at the top there around Haydar, but they do spread across all parts of the tree, you'll see. These are the family member isolates that we found. We had roughly equal numbers in adults and children. Again, they concentrated in two parts of the tree, particularly around Havana and also around Cubana, but again, spread around the tree there and equally between adults and children. These were our 4 animal isolates that we found. They were from a chicken, a cat, and 2 geckos. Thought you'd like that. What I've done here is just plot the animal and the human isolates, and you can see that in contrast to what they found in the Gambia, there clearly is some mixing of where we were finding animal and human isolates and what their genomic clustering was. And I've done the horrible thing to you here. I hope you can see it with the projector, of giving you the code for everything. Again, just to highlight that there was a tendency, there was a mixing of human and animal and environmental sampling in some bits of the tree, but there were additional areas of the tree really where there were further environmental isolates that didn't appear to be appearing in humans or adults. There are a lot of Salmonella out there, I think would be a fair conclusion. I'll move that on before anyone has a migraine. We're going to concentrate on the top bit of the tree here. These are the invasive ST313 strains from our index cases of invasive NTS disease. And the striking and I think important finding here is that we found 2 very close matches from an invasive index case with a family member. In one case it was an adult family member, in the other it was a child family member. We've taken these trees and redrawn them with just the 313 to get better resolution. And you'll see here are our 2 pairs with just a distance of 2 SNPs and 3 SNPs between them, which are closer than any other of the isolates from the rest of the series. So just to summarize, we have conducted this case control study. We've seen multiple serovars across the physical and the living environment, an overall 3% isolation rate, which was similar for all the sites. No isolates from food or water, but we attribute that probably to sampling and methodological issues, so I wouldn't want to write those off. We saw clear overlap between asymptomatic human and animal serovars, but we only found a match with invasive disease with family members, 1 adult and 1 child, and no link with any animals, despite sampling in equal proportion. So this really supports, but doesn't prove, I think, a mainly human reservoir of NTS, and again, supports but doesn't prove a mainly human-to-human route of transmission. Just to emphasise, this is a Salmonella team. We get pretty competitive about our croquet, and that standing in the middle, pointing accusingly, is Ranesh, who did the vast majority of the work on this. He's on an aeroplane to New Zealand as we speak, so he could not be here. And to thank you, this work was initially funded from the core Wellcome Trust funding of MLW to get it going, and we subsequently received a Learning Agenda Gates grant from Bill and Melinda Gates. These are the teams who worked in Malawi and Liverpool to help us. Thank you very much.
That was wonderful. Thank you very much for that. We have time for a couple of questions. I'm going to start purely sampling question here. You sampled geckos. How does that work?
This is easy. Geckos leave fresh droppings on the wall during the night.
So no one's doing rectal swabs
So you can
of
just go in and scrape them off the wall. We were kind of provoked to do this by Jay Hinton, who has done a study into the non-typhoidal salmonella that you find in the snake house in the Liverpool School of Tropical Medicine, and has found an enormous global collection from snakes. And he said, you must look at reptiles.
I have so many more questions for you after this.
Hi, Melita. This is, it's obviously great data, but it's also a little bit frustrating still, because we still don't really know, as you quite rightly say, the mode of transmission of the ST313s. But the evidence is, if you look over longer geographical distances, that these things move through populations. So what's your thought? Where do you think we stand on all of this at the moment?
Well, I think it's if you look at the collective data from Tony, from GEMS, from this, it does seem that when you look, the only place you find is that it's in humans. And I think this migration that we've seen across the continent clearly it seems likely that it's human mediated in terms of migration across the continent. Of course, a study like the one we've done, it could just be that the two individuals in the family had a common exposure. It doesn't prove that they transmitted to each other, but the fact that we are getting such a consistent story over and over does, to me, start to add up that this is a human transmission. Where does that leave us? I guess that it leaves us that there may be a role for kind of hygiene and wash in control, in the same way that there would be for typhoid. So I think in terms of transmission, the sort of ways we might think of it should be more along the lines of how we think of typhoid control, rather than how we would think of control of a zoonotic disease or a foodborne disease.
We only have time for one more question. I encourage anyone else with questions afterwards to chat at the coffee break. Please, you were here first.
Thanks, Melita. Very interesting data. I'm just wondering, obviously, I assume that your ST313 was your multi-drug resistant serotype. Any comment on the drug resistance in your other serotypes, in your animal serotypes? I mean, I know we sell sulfonamide-enriched pig meal, and I wonder if there's similar practices elsewhere in Africa.
So I don't have the full, I knew someone was going to ask me this. We don't have the full antimicrobial resistance data ready, but that which we do suggests that all the animal and environmental isolates and all the non-ST313 isolates were relatively susceptible. There was very little multi-drug resistance outside the ST313s. There was some tetracycline resistance, and tetracycline would be the drug that's used by chicken farmers and so on in Malawi, but there isn't widespread veterinary use in Malawi, I wouldn't say.
Thanks, Melita. And if I took something away from that, it was the bedsock data would suggest that the 6-second rule for drop food doesn't apply in Malawi. Our next talk is from Kudzai Masunda from the City of Harare Health Department in Harare in Zimbabwe, and he's going to talk about knowledge, attitudes and practices related to typhoid fever, the case of Glenview suburb, City of Harare, 2016.
Good afternoon, ladies and gentlemen. I would like to thank Sabin for allowing us to be here and for Abstract to be accepted for this plenary presentation. Ours is a bit of a different study from the ones that have been presented here. We're looking at changing communication and looking at behavior change in terms of typhoid in Harare. So our study setting was in Glenview. It has been among the epicenters of typhoid between 2013 and 2016. And in an outbreak that happened last year in 2016, it accounted for about 36% of the cases. So knowledge, attitude, and practices have been demonstrated as key drivers to the transmission and to the control of typhoid. So what we aim to do was to evaluate the knowledge, attitude, and practices among different strata of the community of Glenview with regards to typhoid fever, and to assess the preferred information, education, and behavior change communication for typhoid. So it was a descriptive cross-sectional study of the residents of Glenview, and data was collected with interview administered questionnaires, observation checklist, and a focus group discussion. Sampling for the interviewer administered questionnaires was done using the homesteads. I think I need to explain here. The homesteads, I think, is the physical address, but we've noticed that in Zimbabwe, at any physical address, there are like 3 or 4 families that are living there. So the households would then be selected using a simple random sampling, and our sample size was 632. For the focus group discussion, we had 9 to 12 participants in the focus group discussions, and they focused on primary school students, secondary school students, adult males, adult females, vendors, and home industry workers. And the tools were piloted in Mbare, and these are some of the data collectors. So we had working definitions of what we described as knowledge. I will not describe them here, good attitudes, and also good practices. And we analyzed our data using EpiInfo, Stata, Microsoft Excel, and Atlas TI, and permission to proceed was sought from the Institutional Review Board of the city of Harare, and also the Medical Research Council of Zimbabwe, and the Zimbabwe Republic Police and school authorities to conduct the focus group discussions, and ethical considerations were observed. This is what we found. So we actually interviewed 635 people, but we discarded 10 questionnaires because they had gross lack of information. And for the focus group discussions with 65 participants, and in our study, the majority of the participants were female, and the median age was 32, and the majority were married and staying with their spouses. And like I said, the median number of households per homestead was 3, and 77% of the people were living on less than $480 per month. We're using the US dollar, by the way. Then 58% were landlords, while 42% were tenants. Then 2/3 said they'd received health education, and the source of the health education was through health workers, through friends, church and IEC material, and through health promoters. And also we had the print and electronic media. Then the reported modes of transmission for typhoid, assessing the knowledge, is that most of the people didn't report the correct modes of transmission. It was only bed hygiene in 51%, and almost a quarter of them didn't know how typhoid was transmitted. And interestingly, in the focus group discussions, some were saying you get typhoid by inhaling the smell from burst sewer pipes. Then ways of preventing typhoid that were cited were also not good. Only above 50% mentioned washing hands with soap and water, and also drinking water from safe sources. Then in terms of the water sources that were termed to be safe, 83% mentioned bore water, and interestingly, municipal water was only mentioned to be safe in 6%. The reported methods of making water safe were mainly boiling water in 63%, and use of chlorinators, the water guard, and aquatabs and bleach. Then overall, 33% of the respondents had good knowledge on typhoid. Then in terms of the attitudes towards typhoid, the perceived risk of self or a family member contracting typhoid was reported to be high in 27%. The likelihood of a carrier transmitting typhoid was high in 22%. The perceived seriousness of the disease, 70%. But interestingly, the perceived seriousness of the disease in Glenview was 42%, and the likelihood of someone dying from typhoid was perceived to be high by 69%. In terms of the prevention strategies, generally, most of them perceive them well, their importance, the overall was 93%. Then the preferred methods of water treatment was mainly the use of water guard, which is 43%, and overall, 15% of the participants that we had a good attitude towards typhoid. Then the practices, 58% use borehole water always for drinking water, and 16% use municipal water always for drinking, and the converse is true. They use mostly municipal water for other domestic uses, and boreholes, you can see only 2% use it for domestic use. And these are some of the cues in the community boreholes that are there in Glenview. Water treatment methods, 62% do not treat their water, and the reasons given are that borehole water is safe, and others are like unavailability of electricity, high cost of electricity, unavailability of the water treatment chemicals, and have someone has not fallen sick from untreated water, and also that the chemicals give an unpleasant taste and smell. For the focus group discussions, they actually confirmed it, saying that underground water, because most of the people use bore water for drinking, underground water is safe because there are no sewer pipes. So, having mentioned that they've got treatment chemicals, on observation, 74% of them did not have the treatment chemicals in their houses at the time of the interview, and about a quarter had the correct tins to store the water and also to draw the water, but they had very good food storage practices. Then the proportion using piped sewer system was 96%, and in terms of waste disposal, it's a mixed method. They use cancel when it's available, they burn sometimes, dispose in open bushes, and they bury. But you can see when we're doing the study, there were lots of pictures like this. I just took only one. Then, in terms of hand washing, frequency of hand washing is high, and before and after activities, like common activities, but frequency of using soap with water to wash is low. Then, in the discussions, what you notice is that there's a generally low level of knowledge in terms of good attitudes, good knowledge, good attitudes, and good practices regarding typhoid in Glenview. And the majority of our participants were females, we think, because we collected the data during the day and also during the week. That was Monday to Friday. Then there's an average of 15 people per homestead. And in this area, most of the houses are 3-bedroomed, and people are living with their spouses, so it translates to like 1 bedroom per family. So there is overcrowding problem there in Glenview. Then we thought, because they had outbreaks for a long time, and also because at each outbreak we go and give information. Glenview, by the way, is also the area that had the big typhoid outbreak in 2008 and 2009, but there was generally low level of knowledge on typhoid. So we're thinking that we need to have strategies and modalities to deliver this health information. Then, the State of Harare message has been that since 2010, because we are not sure also of our water sources anyway, that we need to treat all drinking water regardless of the source. So we're strict in terms of analyzing what people think about the water in Harare. So only 4% of the people mentioned that no water is safe. So it means they're thinking most of the water they're drinking is safe, and thus are not treating it. Then we have already seen that some of the boreholes in Glenview, where the people are taking water for drinking, are contaminated, but 83% of the mentioned bore water is being safe, and that they use it always for drinking, and that they are not treating it. Then we also thought that since the majority of the participants are women, who are generally the caregivers in the homes, and also that they are living below poverty datum line, studies have shown that poor communities and where there are women, they've got higher perception of infectious diseases, but in our cases it was low. Then in terms of hand washing, everything was good, the knowledge was good, the perception was good, and actual practice was good. What was not good was that they are not using soap for hand washing. Okay. Then the limitations was that it was social desirability bias, because some of the questions people would they know the correct things, but they may not be doing them. So some of them may have been telling us the correct things, but not what they're actually doing. And also, I think I already mentioned that we did the study during the day and also during the week, and we had more females than males, so that could have brought an element of bias. So the proportion of good knowledge and good attitudes and good practices was low, and also the practices that negatively impact on prevention and control typhoid were identified in this study. These are the recommendations that came out of the study. We need more health education strategies to improve social and individual behavior change. We need to promote the use of water treatment, fit inline chlorinators to our boreholes, and further studies to determine the prevalence of carriers and to determine risk factors in these areas. And then because we had prior knowledge of the resistance pattern, we also think we need to check on the resistance pattern in this area. These are my acknowledgements, the study participants, UNICEF for funding us, and also the research assistant and data collectors, and our biostatistician. Thank you.
Thanks, Musa. And we've got some questions. Thanks.
Hi, Rob Bryman from Emory. Thanks for your presentation. So how would you specifically change your messaging and your methods to have impact? I don't think what you observed is limited to your site. I think it's a problem everywhere. Do you have thoughts about changing strategies?
Okay, yes, they are. What we have since done, we've developed new communication materials, because the ones that we had were from the 2008 cholera outbreak. So we're thinking maybe they are not giving the correct information on where we are. And also, most of our health promoters are aged and may not be delivering the messages that we want. So we are now trying to change the health promoters that we have so that we have the younger ones and who can deliver the message that we want.
Sam, karaoke.
Thank you very much. Thank you, Dr. Masunda, for that nice talk. I always say once a microbiologist, always thinking that line. So I don't know, did you get these strains analyzed in Zimbabwe? Do we know what is causing these outbreaks in terms of serotype or antibiotic resistance patterns and so forth?
Okay, what I know is the strains, we have sent them to the National Reference Laboratory and they send them to South Africa for analysis, but I do not have the results.
Okay, because we would actually be willing in our consortium to really help in this analysis, maybe a bit later on.
Okay.
That's very good. Thanks, Sam. Yes, Rob?
In your presentation, you focused on water, but my memory of the markets in Harare is that often they use water to make vegetables look more attractive and to wash them and so forth, and that water may come from anywhere. So have you also looked at the practices in relation to vegetables and whether they're cooked and so forth?
Okay, this study came after we had done another study where we were looking at exactly the risk factors you're talking about. But that study, we had done it in Budiriro, and it had shown that people who eat from the vendors, which is uncooked and were male, were risk factors for getting typhoid. So we now wanted to assess the knowledge levels of the people who were in Glenview, just to see whether our communication strategy is the correct one.
Thank you very much. Thank you very much. That was great. Our last speaker of this session is Vittal Mogasale from the International Vaccine Institute, who's going to discuss spatial and temporal patterns of typhoid and paratyphoid fever outbreaks, a worldwide systematic review.
Thank you. Good afternoon. Thank you, Dennis, for inviting me to present. Thank you, the reviewers, for accepting the abstract. So I'm going to talk about the spatial and temporal patterns of typhoid based on outbreak review. So this is the outline of my presentation. And we did a systematic literature review. So why did we do this? Why did we do this? So outbreaks are not often included in the disease burden studies because the disease burden studies often come from the extrapolation from surveillance studies. And the outbreaks would be included in the surveillance only if the outbreak occurs during the surveillance period in the catchment area. If anything happens outside the surveillance area that may not be included. And often outbreaks are not reported because they are not collected systematically and not reported. And this kind of outbreak information is helpful in geospatial and prediction modeling, because when it's linked to the GIS information, it can be used. So we used PRISMA guidelines, and we used two kinds of databases, medical database like PubMed and Embase, and we also used epidemiology-specific database. These are like reporting the news of outbreak with some verification, GDON and ProMed Mail. And the period of the review included from 1990 to 2016, December. And this report, we use the English language, but often in some of the databases, the outbreaks reported in other languages is translated to English. We used whatever is available in the reports. And outbreak definition is based on the as authors reported. We tried to validate the outbreak reports with the WHO definition, which we couldn't do, so we took by face value. And any method of diagnosis, culture, or the serology, or clinical, whatever is the methodology used in the outbreak, we used all of them with the face value. So this is the search term we have used. Duplicates were removed, and then we identified the unique outbreaks, because many of these reports include, like 2 reports may include same outbreaks. They can be reported at the same time. And sometimes same report includes more than one outbreak. In that case, we have to identify the unique outbreak based on time, place, or wherever the reporting period here. And it's linked to the GIS information based on the Google map. And then we had a predefined data extraction, and the 2 researchers did the work. And this is the PRISMA flowchart. And if you see at the end, after all this review process, we had 256 papers, 256 papers with 226 outbreaks. This is the map showing where the outbreaks occurred and what time period. On the y-axis, you will see the different regions, Africa, Asia, Europe, different region, and in the x-axis, you will see the time period starting from 1990 to 2016. And the size of the circles suggests the size of the outbreak. And you can see the lowermost, Africa and Asia, there are a lot of outbreaks happening, large-sized outbreak, almost every time period. And these are the numbers, how many numbers of outbreaks we identified in region, and what is the total size of the cases, number of the cases, with the variation in the size of the outbreaks. And then based on the GIS, we plotted the outbreak on the map. This map shows the typhoid and paratyphoid outbreaks. And the typhoid outbreaks are in red, paratyphoid outbreaks are blue. And you can see the more blue spots in the Asian region. And these maps represent what was the diagnostic method used in determining the outbreak. The blue one is used by the blood culture. And green one, the method is unknown. Sorry, the green one is the Vidal test was used, and the red one is the method is unknown. It's not defined how they identified the outbreak. Multi-drug resistant outbreaks, you can see the blue dots represent the multi-drug resistant outbreaks, whereas the green are wild type and red one are not specified what kind of outbreaks. And then we looked at the outbreak, what are the reported causes of the outbreak, and of all the outbreak, only 40% reported at least the main reason, or one of the reason is contaminated water. But most of them did not report what is the cause. And the foodborne is around 15%, and person-to-person transmission is 1%. There were also the cases, outbreaks reported based on the imported typhoid fever. And when we looked at the underlying reasons for the water-related transmission or the outbreak occurrence, we classified into 3 categories: failure of infrastructure and planning, which is the pink big circle, educational, culture, and social issues, and the environmental damage, which is dashed line, the circle containing. So they all overlap. So we represented it in the Venn diagram, showing how they all overlap, because many outbreaks had more than one underlying reason. So when we look at this geographical location of these outbreaks, they represent many of the regions which we consider as high endemic for typhoid fever. And from the outbreak review, we understand that contaminated water is main underlying reason for the outbreak. And we see that many of the places, particularly Asia, has both typhoid and paratyphoid outbreaks happening. So at least bivalent vaccine would be more useful in Asian region. The limitations of our study: these outbreaks are author-defined. We could not verify it against WHO definition. And we have to understand that different health systems have different capacity to report outbreaks. Maybe the health systems in Europe or North America report outbreaks more often than the developing country health system. And also, in places where typhoid is known to be endemic, in that situation, outbreak may not get attention and may not get reported. Yeah, then the different databases have intrinsically built reporting bias. Some databases capture more information from the particular geographical regions. That reporting bias causes some bias in the overall findings. In terms of conclusion, enteric fever outbreak burden remains high in endemic low-middle income countries. And we should account outbreaks in prioritizing our decisions, public health decision, vaccine introduction decision, policy decisions, and as and when possible, as appropriate, the available vaccines could be used for outbreak controls. And for the detection and reporting of outbreaks, we need standardized method so that in future the outbreaks are comparable. This main work was done by Samuel Kim, who is a medical student in Imperial College. He came to Seoul for 3 months to work with me, and because of his efforts, we could do this work. And co-authors are listed here. Thanks to co-authors. And this did not receive any specific funding, but IVI receives core funding from Government of Korea and Sweden. Thank you very much.
Thank you. That was wonderful. We have time for a few questions. Yes.
Thank you, Vittal, for that very important information. And you raised the issue that water contamination is an important point. And taking the clue, I will ask the question to one of the previous speakers, Tahir, who reported outbreak in Hyderabad, Pakistan. I think when outbreaks are occurring, we should also look for not only typhoid, but hepatitis and diarrhea. So my question to Tahir is, was adult hepatitis cases and diarrhea cases also seen, are being seen during Hyderabad epidemic? Because these are important. If we have to interrupt the epidemic, unless we are able to find the source, we won't be able to do that.
No, we didn't see adult patients of diarrhea.
And hepatitis E?
No.
Great. Other questions for our speaker?
Thanks, Vittal. I'm sorry, I have a sore throat. I have one comment and a question. The comment is just to commend you on this review. It's going to be immensely helpful for the WHO policy process, as we've been discussing already. And we're also looking forward to getting additional information from colleagues in CDC who have more detail on actual outbreak response, etc. So looking forward to working with you on the additional work. My question is, I didn't see any data on the three parameters I'm thinking about now. Fatality during the outbreaks, duration of the outbreaks, because there have been some publications of very extended outbreaks, particularly in the African setting that I'm familiar with, and response in general, outbreak response in general. And I'm just wondering whether it's because you didn't have these data uniformly across the review that you didn't include them, or do you have a sense of how often those three parameters or outcomes in general are being reported?
Thank you, Ajowa. You have rightly guessed the answer. We originally wanted to collect the fatality rate. It was part of, in our protocol we had included, but when we got the data, when we were reviewing the results, there are not many outbreaks that reported case fatality rate because they were reporting somewhere in between the outbreak. And for the same reasons, we also couldn't get the duration of outbreak because most of the reports are coming in the middle of outbreak. For that matter, the size of the outbreak, what we represented, may not be correct, maybe underestimation because many of these reports came in between outbreak. Same reason we don't know the outbreak response. We haven't tried to extract that data, whether the response was done and what was the response. Thank you.
Any further questions? Yeah. If not, I'd like to thank all our speakers. Everyone did a wonderful job. And it looks like there is another tea and coffee session upstairs with poster presentations. So please enjoy.